Our next speaker is Dr. Ashokkumar Balasubramaniam. Dr. Ashok is an associate professor at the School of Biotechnology from Madurai Kamaraj University. His research focuses on genetic disorders of nutrient transport and metabolism, diabetes, and microbial degradation of xenobiotics. He has published over 110 papers, authored 15 book chapters, holds multiple patents, and received awards including the Tamil Nadu Scientist Award in 2021 and ICMR International Fellow Award. Today, Dr. Ashok will talk on uncovering the burden of riboflavin transporter deficiency among Indians, metabolic insights and emerging strategies for gene therapy. Thank you, sir. Over to you.
Thank you. Thank you so much for the nice introduction. And at the outset, I express my sincere thanks to the organizer, particularly Dr. Nihal Thomas, for inviting me to share my research expertise in the field of genetic disorders related to nutrient deficiency. And for today's talk, I have chosen riboflavin deficiency. Right. For a simple analogy to introduce about the subject what I'm going to talk about, I've given this image. From this image, you can understand that there are three different gods from the three popular religions I have pitched over here. And from this image, you can understand that they are gods, right? So, who are distinguished by just by providing bright yellow halo behind their heads. So, we believe them, they do have a kind of a power. With the power, they will be able to create a life or they will be able to take care of us. This is what we do believe normally. And what is that bright yellow halo? This is the question everybody is asking. So some people, physics people, they have found an answer and they are saying that it is a God's own magic particle, which is nothing but Higgs boson particle, right? So if a chemistry person, if you ask a chemistry person, they will say something different, okay? Right. Now, some research in the biology, they have come to know that it is a kind of a simple micronutrient which is present in every single life where a life is originating. For example, if you take an egg, from the egg only a life is originating, no? The egg yolk, which appears yellow in color. What is there? What is that? If you really want to know about it, I have some answer for that. The compound which is giving bright yellow coloration is nothing but riboflavin. Riboflavin is one of a micronutrient which is coming under the category of vitamins. Vitamins are generally classified into two categories. They are water soluble and fat soluble vitamins. So this riboflavin, which is simply known as B2 vitamin. Okay, right. So look out the image which is there in the bottom. I'm showing you here with the earthworm. In the earthworm, particularly you look into the region 2, which is nothing but a clitellum. So suddenly, if we think about earthworm, immediately something will coming up in your mind that, so this has a potential to regenerate. Okay, if you cut this worm, okay, so it has a potential to regenerate. But the question is that where you have to. So if you cut it just behind the clitellum, next to the region 2, if you cut it next to that, this worm will not regenerate it. Okay, right? So you can see that the clitellum region seems to be distinctly, which is appearing yellow in color. Recent research have found that these clitellum where huge number of stem cells are there, these stem cells which are accumulating higher concentrations of riboflavin. Why it is accumulated over there? The key point is that for any cell replication and its duplication, we need a lot of energy. For providing these energy during metabolism, in the case of oxidative phosphorylation, which is happening in the mitochondria, right, there is a component which is produced out of riboflavin is essential, which is nothing but two cofactor. They are FMN, flavin mononucleotide, and flavin adenine dinucleotide. These two are cofactor which is coming out of riboflavin. Because of that, only energy yielding pathway which could produce lot of ATP in the mitochondria. That's nothing but our powerhouse, right? So this energy only is being used for the cell division and multiplication. Simply, I can say that vitamins which are essential for any cell division and its growth. Without vitamins, especially water-soluble vitamin, there is no cell division and growth. In the next slide, you can see that cancerous stem cells were imaged. These cancerous stem cells, which are autofluorescent in nature, while they dissect, there also scientists found that riboflavin, which is accumulated like anything. So that riboflavin, if you expose it to UV, it will be fluorescing. Okay, right. And this is the pure riboflavin color, bright yellow color. Flavin, which is nothing but which denotes yellow, right? And there, when you expose it to UV, riboflavin will be fluorescing. You can see the bottom. You can see the egg yolk from a local breed and the broiler one. The local breed yolk, which appears very bright in color because of the huge concentrations of two important vitamins. One is carotenoid, vitamin A, and the other one is riboflavin. Because of that, the egg yolk is brightly colored in yellow, right? So there in the last image, I am showing an egg of a female human egg. There also you can see that there is a humulus region, a layer which is surrounding the egg, which is also appearing yellow because riboflavin is accumulated hugely in this region. In all these cases, you can come to know that, okay, wherever cell division is taking place, in those cells, particularly the stem cells, riboflavin is accumulated in higher concentrations. This is the point I really want to stress upon from the last two slides. So riboflavin is very, very essential for any cell growth and its development. Right. So some biochemistry related riboflavin I really want to highlight over there. So it is a water soluble vitamin which is shortly known as B2 vitamin. So this riboflavin is biologically inactive. Once it is getting transformed into a cell, this riboflavin will be converted into either FMN and FAD. These two are biologically active molecules which are known as cofactor. There are so many, approximately 80 different enzymes are there in the human cells. They do require either FMN or FAD as a cofactor. Without these cofactor, those enzymes cannot function. The cofactor, the enzymes that depend on either FAD, FMN are known as flavoproteins. Flavoproteins are nothing but proteins that they depend on FMN and FAD as a cofactor. They play very crucial role in the fatty acid beta oxidation, amino acid metabolism, TCA cycle, which is the key pathway in the energy yielding pathway, TCA cycle, Krebs cycle, and even in the electron transport chain. In all these cases, FMN and FAD act as a cofactor. Riboflavin is the only source which can be converted into FMN and FAD. The chemical structure of riboflavin is shown over there. That consists of ribotyl side chain, which is nothing but a reduced form of ribose sugar, and which is also added with isoalloxazine ring, that gives yellow color. If it is added with one phosphate group, that's nothing but FMN. If it is added with adenine nucleotide, it is said to be FAD. Right. So human body does not have the ability to synthesize riboflavin. Not only riboflavin, most of the water soluble vitamins cannot be synthesized by our body. Vertebrates have lost the ability to synthesize. We depend on the food sources, the exogenous sources, that food whatever we eat, that food that should contain riboflavin, which is getting absorbed in our intestine. In the intestinal epithelial cells, in its membrane, there are proteins known as a riboflavin transporter, which are facilitating entry of these riboflavin into the cells. So intestine is the primary portion where the major absorption is taking place. So through the intestinal epithelial cell, the absorbed riboflavin, which is entering into the cell, and it will be mixing with the blood. Through the blood, this will be delivered to every single cell of your body, from brain to toe. So this is how the transport mechanism which is working over there. For that, you should have riboflavin transporter. Naturally, this riboflavin, which is present in higher concentration in the leafy vegetables, milk, cheese, almond, legume, mushroom, egg. Not only in the vegetarian items, it's highly rich in the non-vegetarian meat, especially in the liver. Huge concentrations of riboflavin which is present in the liver. So if you take a good diet, in a sense, we will be able to meet out our body requirement. Right. So what is the role of this riboflavin in the mitochondria? See, in the mitochondria, when you look at the electron transport chain, there are 4 complexes, 5. Complex 1 to 4 and complex 5, and there are other shuttlers like coenzyme Q10 and cytochrome C. All these these proteins which require FMN or FAD as a cofactor. If these cofactors are not present, in a sense, electron transport chain cannot be functioning properly. If it is not really happening, in a sense, ATP cannot be produced from one single glucose molecule. We know that it has been already calculated. 36 ATP is produced from out of one single glucose. That may not be possibly produced if FMN, FAD is not there. If it is not there, in a sense, this will be inducing oxidative stress to the mitochondria that will cause destruction of your mitochondria. If this destruction is happening to the neuronal cells, in the neuronal cell, if it is happening, it will lead to neurodegeneration. You can see over there. So how a neuronal cell, if it is losing the level of FAD and FMN, finally it will be ending up with neurodegeneration. All right. So I already told you that there are riboflavin transporters in the human. There are 3 different riboflavin transporter. They are SLC52A1, riboflavin transporter 1, SLC52A2, riboflavin transporter 2, SLC52A3, riboflavin transporter 3. Okay, you can see that their localization in a cell in the human body is different. For example, riboflavin transporter 1 is usually expressing in the placenta and the small intestine. But at the same time, riboflavin transporter 2, which is hugely expressing in the brain. So it is known as brain-specific riboflavin transporter. But A3, riboflavin transporter A3, which is intestinal-specific riboflavin transporter, because this is usually expressing in higher concentration in the intestine. So that is the key riboflavin transporter, which is responsible for the internalization of your riboflavin into the intestinal epithelial cells. If there is a mutation in the riboflavin transporter 2 and 3, you will have neurodegenerative disorders. If there is a mutation in the riboflavin transporter 1, which is not responsible to cause whatever the neurodegeneration that I talked to. So key riboflavin transporters are brain-specific riboflavin transporter 2 and the intestinal-specific riboflavin transporter 3. So the secondary structure of the proteins have been predicted. They do contain 11 to 12 different transmembrane domains. Right. So this is the overall image which I have published in the year 2017. So what is really happening in the intestine? How the riboflavin transport is taking place. In the epithelial cells, you have finger-like projection, microvilli. In the microvilli, you do have an expression of riboflavin transporter 3 abundantly in the case of intestine. Through that, riboflavin is coming inside along with the proton. It's a proton-coupled riboflavin transporter. Okay. Now it is coming inside. Once inside to the cytoplasm, it will be immediately converted into either FMN or FAD. Then this FMN and FAD will be transported into mitochondria. We are not sure that still whether riboflavin could go into the mitochondria or not. So FAD, FMN can go into the mitochondria and they will be able to work in the electron transport chain. And in the bottom, you do have riboflavin transporter 2 and riboflavin transporter 1. They will be facilitating their exit in the bloodstream. Through the blood, it will be it is entering into the either brain cell or kidney cell or liver cell, wherever it may be. Okay. But in the case of brain, you do not see the expression of riboflavin transporter 3. You have an expression of riboflavin transporter 2. Because this is the brain-specific riboflavin transporter. Mutation in the riboflavin transporter 3 and 2 only causing riboflavinopathy, which is nothing but neurodegenerative disorders that are associated with the riboflavin. Right. So this I have already talked about it. What is really happening from glycolysis to ATP yield in the case of mitochondria. Right. So what are the diseases that are associated with the riboflavin deficiency? Commonly, these are all certain disorders like preeclampsia, cataract, cardiovascular disease in cancer, So their deficiency may be related with these disorders, but particularly there are few genetic disorders which are associated with them. The name of the genetic disorder is that brown-violet-van Leer syndrome, shortly known as BVVLS, and the another one is Faziolon syndrome. the symptoms of these diseases which has been highlighted over there, people will be experiencing problem in breathing, swallowing, hearing, and they do also express symptoms in the movement, gait phenotypes, you will see over there. So if you have a mutation in the riboflavin transporter 2, that deficiency is shortly known as RTD2. If you have a mutation in the riboflavin transporter 3, that disease is known as riboflavin transporter deficiency 3, that is RTD3. What is the difference between phaseolon and BVLS? Both entities are almost the same, except only one phenotype. Deafness will not be there in the phaseolon syndrome, but deafness will be there in the BVLS. That's the only difference. We do not know why specifically deafness is happening in certain cases, why it is not. Still, it is a puzzle that need to be answered. But another disorder which is related with riboflavin transporter 1, which is nothing but multiple acyl-CoA dehydrogenase deficiency, which is related to the fatty acid metabolism. But riboflavin transporter deficiency 2 and 3 only, which is causing neurodegenerative disorder. The symptoms are swallowing difficulty, muscle weakness, respiratory difficulty, facial dysplasia, and tongue atrophy, dysphonia, facial weakness, reduced reflexes, and premature death. There are so many major symptoms which are associated with the disease. Some of the images of the patients which have been documented with the BVLS and the phaseolon is shown over there by Green et al. In the year 2010 only, this group only identified riboflavin transporter is responsible for causing this disease. By performing clinical whole exome sequencing, I happened to visit to his laboratory, which is there in the University of California, UC London, which is there in the London. Okay, I went over there to get experience in clinical whole exome sequencing. Right now, at Madurai Kamaraj University, I have established this diagnostic facility, and we are performing clinical whole exome sequencing also for the patient samples. And right, so far, what are the mutations have been documented? Two of my PhD scholars, they have already completed, and they have just drawn these images. These are all prevalent mutations in the riboflavin transporter 1. In the riboflavin transporter 2, these mutations are quite commonly observed, and in the case of 3, so many mutations have been documented, especially in the case of exon 2 and 3. Right. So, what is the burden of neurological disorders among the Indian population? If you look out at, there are very common neurological disorders, but motor neuron disorder prevalence is very, very less. So, these disorders, whatever I have talked about it now, these disorders are known as rare genetic disorders in India. There are almost 7,000 to 8,000 genetic diseases have been documented. But in the case of India, only 450 cases with genetic disorders, rare genetic disorders have been documented. But, their prevalence, their burden in our Indian population is not yet uncovered. So, I am primarily working on uncovering the burden of these genetic disorders in Indian population, especially South Indian population. Right. These are all certain patients whom we have enrolled in a study, and the genetic analysis part have been already completed, and all these cases were already published. Okay, some of the cases images that I'm showing over here. You will see weakness in the limbs, both the hand limbs and leg limbs, and gait phenotype you will see, tongue atrophy normally you will see, and the facial weakness also you will see in these cases. There is a particular case. There are 320 families which are living near Tamil Nadu and Andhra Pradesh border, near Nellore. The name of the disease is Madras motor neuron disease. The name of the disease itself, Madras motor neuron disease, but the genetic cause for this disease is not identified so far. Already I have performed clinical whole exome sequencing in the people along with the collaborative research with the CCMB, but still we are not able to identify the culprit gene which is responsible for this particular disease. The name of the disease is MMND, Madras motor neuron disease. All right. So these are all certain cases. We have gone for exon based sequencing. If not, clinical whole exome sequencing we have performed, and we have identified mutation in these cases. And the chromatogram that I'm showing over there, once after identifying those mutations, we do generate, the wild type and mutant construct along with the fusion of GFP. And these constructs which were expressed over there in the cell lines like HEK293 cells, and those cells were observed under confocal microscopy, there we have imaged whether, since they are localizing in the membrane now, you should see the expression over there in their membrane. Okay, so you can see that if you transfect with an empty vector, how they will be appearing, and if you transfect with the wild type transporter, if it is with a mutant transporter, you will be able to see the differences. When you compare with the wild type, you can see there is a leaky expression in the case of riboflavin transporter in the membrane of the cells which were transfected with that. That itself tells that the riboflavin transport efficiency of the mutation is highly diminished. So this could be the major cause for the neurodegeneration in the patients. So this is the outcome of confocal microscopy study what we have performed, and at the same time we have deeply looked into those cells which are expressing these GFP. So you see normally apical side and the basal side, basolateral side expression of these GFP constant. But look at the wild type, you see the expression all over the cell, but in the case of the mutant, you see expression only in the apical side, not in the baso and basolateral side. So there is a diminished expression we have observed with a particular case, and we highly concluded that the expression itself is a problem in the case of this mutant. Because of that, the patient is having riboflavin deficiency. Ultimately, he developed a brown-violet van Leer syndrome. Right. So this is another unique case which we have found from our country. One of the Indian patient who was found to have mutation in both the riboflavin transporter. We have even published a paper like that. First case who is having mutation in both the riboflavin transporter. This is very, very unique, which is not usual. Either you will see mutation in the riboflavin transporter 2, if not 3. But we have mutation in the both the cases, riboflavin transporter 2 and riboflavin transporter 3. And from this chromatogram, you will be able to understand both the mutations which are present in the A2 and A3. All right. So once after like that, before performing, the functional study, we just constructed both the wild type and the mutants in the mammalian expression vectors, and we performed the transfection with the cell lines, and we really quantitated the efficiency of riboflavin transport in the cells. How we did that? We have radio-labeled the riboflavin, and the radio-labeled riboflavin was used for our experiments. After a certain time of incubation, the amount of riboflavin which is accumulated within the cell was counted using scintillation counter. So how much of riboflavin has been taken up by the cells has been counted, and it is compared with the wild type. So from the observation, you can see that when you compare with the riboflavin transporter wild type 1, there is a diminished riboflavin transport by the mutant P144T. P has been replaced with the T in the case of SLC52A2. But in the case of SLC52A3, you can see that there is a complete depletion of riboflavin transporter efficiency with the case of N21S. At the amino acid 21, asparagine is mutated into serine. So this is the mutation we have got in an Indian patient, and we performed this functionality assay using radio-labeled riboflavin. We concluded that the riboflavin transport efficiency is completely affected because of the mutation. And again, for this mutation also, we performed confocal microscopy. It's a wonderful image. You can understand very clearly in the bottom image. You can see that GFP, HRFBT3, you can see the membrane itself, which is clearly visible over here. And we have used endoplasmic reticulum DS red as a marker that will be staining only the endoplasmic reticulum. There is no overlay, and clearly you can see the membranous expression of riboflavin transporter 3 in the case of wild type. Look at the bottom. Okay, you see the complete GFP protein is internalized within the cell cytoplasm. There is no expression at all in the case of membrane. There is no staining in the membrane. That indicates clearly that the synthesized mutant riboflavin transporter, which is accumulated primarily within the cytoplasm, which is not going to the membrane. There is a problem in the trafficking and the targeting of the riboflavin transporter. There are so many motor proteins which are associated with the transport and delivery of these membrane proteins to their membrane. If there is a problem, if the mutation is not able to help for its targeting and trafficking, then obviously this is the phenotype you will see. So the patient was completely deficient to riboflavin in his blood. The level of riboflavin was completely less. Because of that, he developed brown-violet van Leer syndrome. This is the outcome of this wonderful functional studies what we have done at the MKU. And these are all two papers we have published. You can see first genetically proven case with the mutation in 2-riboflavin transporter. This is what we have published in the case. And there we have published a unique phenotype we have observed. First time pseudo-dominant inheritance with relevant to the disorder we have been identified. Normally it is it's happening under recessive condition only. The first time phenotype, pseudo-dominant inheritance we have recorded. That also we have published in the European Journal of Neurology. All right. So actually this study is not done at my laboratory, but I really little bit want to talk about gene therapy. So how to rescue these patients? So very simple thing you can do through supplementation orally. You can provide oral supplementations of riboflavin. That's enough. Through the leakiness, though you may have mutations, the certain quantity of your riboflavin which will be entering into the cell. So you should not give very less quantity of riboflavin in this case. 1000-fold higher riboflavin what your body that should be loaded over there. That will be helping for the entry of some quantity of riboflavin because it's a micronutrient. You need this riboflavin in your blood at a nanomolar level. But when you dump it with the huge concentration, some quantity is really possible to get into the cell, and that's enough to protect you. The patients who are having this riboflavin deficiency can be supplemented orally with riboflavin, and you will be able to save the baby. Otherwise, the symptoms will be worsening and it may end up with a death. But you can save the baby by oral supplementations. So this is right now the therapeutic strategy for this, but this is not a permanent therapy. Okay. So in addition to that, for complete therapy, gene therapy holds promise. Okay. So May et al. Recently has published a paper in the journal Frontiers Cell Neurosciences in the year 2025. So some of the images I would like to show because of the time constraint, I'm not able to, but just I will quickly move on it. Okay. So they have developed, from the iPSCs, neuronal cells they have developed, and then later they could also measure the length of the neurons, neurites length they have measured. And after doing gene therapy, and they are also measuring, and they are showing that there is an increase in the length of the neurons after delivery of this gene with the help of adeno-associated virus 9, and they were able to rescue the phenotype whatever has been lost. And with that, I will just conclude my talk. And the collaborators who are really worked in this work, Dr. Vaikundaraju from Indira Gandhi Institute of Child Health, Bangalore. He is the one who shared a patient blood sample. So far at the facility of Madurai Kamaraj University, I have diagnosed 40 cases with BBVLS. So in the world, so far only 220 cases. So from our country, I have contributed 40 cases we have diagnosed at our facility. So this is really a worrying part. We need to think about it. The major reason why the prevalence of this disease is high among South Indian population, consanguineous marriages is one of the major reason for that, and the poor education. So I'm talking in various groups to create awareness about this congenital disorder. Professor Henry Holden, who is the one who is working there in the University College London, who provided me training in the clinical whole exome sequencing. Professor Hamid M. Syed, my postdoc boss, under his guidance only, I just learned the techniques how to perform vitamin transport physiology. I have already performed all these techniques for thiamine, riboflavin, folic acid, and so on. Right. And Saravanan Madheswaran, my student, now he is a faculty at IIT Kanpur, and I'm also collaborating with him. Dr. Varun Venkat is a colleague of Dr. Vaikundaraju. Dr. Varalakshmi, my colleague at Madurai Kamaraj University. And DBT is majorly funded by CSCR, DBT, UGC, and ICMR funded me. So far, I'm sanctioned with 21 research grant. And thank you. With that, I conclude my presentation. If there are any questions, please feel free to ask me.